I would like to introduce Kelvin Liu, who is the CEO, founder, and intrepid leader of Circulomics here. So today he's going to talk about a few of the really awesome, interesting projects that we have going on here. So with that, it's all you, Kelvin.
Thanks for the intro, Jeff. All right, so I'm gonna talk a little bit today about kind of how we approach sample prep, and as an example, I'm just gonna kind of walk through two projects that we've done recently. One of them is on PacBio, one of them is on Nanopore, and then the different ways that you approach sample prep in those projects. So all right. So are we on slide 2, Jeff?
Yes.
Okay,
full screen.
Yeah.
Okay, there you go. Wait, do you guys see full screen, or do you guys see the slideshow right now?
We don't see the slideshow. Yeah, we see it's not in presenter mode.
Wait, you see just the slide, or you see the little slides on the side? Sorry.
We see the slides on the side. Sorry. Yeah.
Okay, it's on.
Here's switch monitors.
It's sharing. I got 4 monitors here, so it's okay. Here we go. We got the right one now.
Perfect.
Okay, yeah. So probably don't have to belabor this point too much with this crowd, but obviously, it's in order to get nice sequencing results, it's important to get good quality DNA going in. So if you want nice long reads, nice throughputs, good quality reads, the DNA going in obviously needs to be big, needs to be pure. It shouldn't have a lot of contaminants. They're going to inhibit either the library prep or the sequencing, and then also needs to have low damage. But one point that might be a little less obvious is that the library prep is also important, right? So it doesn't matter if you put in really high quality DNA going in. If it gets kind of mucked up during library prep, then your sequencing results are going to be compromised as well. So when we talk about long read sequencing in particular, this is even a little bit more challenging, because here you need really clean high molecular weight DNA. So this kind of very clean high molecular weight DNA can be challenging to get with traditional spin column and magnetic particle methods. So the methods that actually generate the highest quality DNA are generally precipitation and agarose plug purification. But these methods can be pretty slow and tedious, particularly agarose plug purification. So basically what we've developed is kind of a technology that we've developed here at Circulomics is called Nanobind. The easiest way to think about this is they're small magnetic disks. So in principle, they work very similar to magnetic particles, except instead of having millions of particles per tube, we have one big disk. The disks are typically about 1 millimeter to 5 millimeter in diameter. If you take an SEM of the surface of the disk, you'll see that there's all these microscale wrinkles. And then if you look on top of those microscale wrinkles, you'll see that there's all these nanoscale wrinkles and flakes. And what that does is this gives you a very high surface area to bind lots of DNA, but it also protects the DNA from being fragmented. So we're able to extract super high quality DNA, basically DNA that's up to megabase in length, using a simple bind, wash, and elute protocol. One other difference between what we do and a lot of other kind of more general sample prep companies is that we're really focused on the kind of the long range genomic market. So we do a ton of sequencing internally to make sure that all of our platforms, all of our DNA works well on Oxford Nanopore, on PacBio, on BioNano. So the core of our technology, our platform and our products are definitely built around our Nanobind technology. But as I mentioned earlier, it's really important that every step in that protocol, everything that happens to the sample before it goes onto the sequencer is performed well, because any part that doesn't go well is going to affect your sequencer results. So what we're really trying to build is really a comprehensive sample prep portfolio that takes care of everything from high molecular DNA extraction to size selection, library preparation, and also allows you to do that in either a manual or an automated format. So on the left here, you can see what are our Nanobind big DNA kits. So basically with these 3 kits, you can extract high molecular weight DNA from a pretty broad variety of samples. We're continuously expanding the chemistries and protocols that we offer here. So you'll see updates in the next year to our CBB kit, also to our plant kit, and also to our tissue kit, so that you'll be able to extract anything from plants to cells, bacteria, to tissues, from mammals, from reptiles, from insects, worms, fungal samples. Be a pretty broad range of stuff we can get high molecular weight DNA from and generate beautiful sequencing results from. On the right side, we have some of the products that we're going to put out in the near future. So the first kit that we're going to release shortly, which will be the subject of the second half of this talk, is our ultra long library prep kit. And this is a kit that you can use to generate ultra long reads on Oxford Nanopore. We'll also be putting out an automated extraction kit. So all of our extractions will be able to run on Thermo's Kingfisher instruments. The first kit will be for cells, bacteria, and blood, and this will basically enable you to do high molecular weight DNA extraction from a plate of 96 samples in 30 minutes to an hour. The sequencing performance is identical to our manual processes, so you can expect to get 30 to 50 kb and 50s across an entire plate of samples. And then we also have other kits that we're working on as well, including a metagenomics kit. Aside from our Nanobind products, we also have our short read eliminator products, and these are used for rapid size selection. So basically, you can use these kits to deplete low molecular weight DNA from your high molecular weight DNA sample. The kits use a very simple precipitation process that takes about an hour. We have 3 different kits with different cutoffs depending on the quality of your input DNA and how much enhancement in your sequencing read lengths you're looking for. But typically, we can see like a 5 to 20 kb enhancement in your read length and 50s when you use these products. So the first product, the first kind of project I'm going to talk about is a project to sequence a Vol sample. So Polly Campbell from UC Riverside and Brian Cougar, who's now at Brigham and Women's Hospital in Boston, they approached us about this Vol they wanted to sequence. So they had a few individuals they wanted to sequence, and these are one of the samples was high quality sample that they had high quality fresh frozen tissue from. I think this was an individual that's actually from that was raised in their lab. But some of the other individuals they were looking to sequence, which is often the case with non-model organisms, were caught in the wild. So they had one individual that had been caught in a trap, and that individual had likely been there for maybe a day or two. So it was a little less fresh than the fresh frozen sample. And then there was another individual that had been in the trap, obviously, quite a few days. So in this case, when you're looking at these non-model organisms, you often don't have the flexibility to choose. What you get is what you get. So you kind of have to make the best of what you have. So these were from three different individuals. All of them need to be sequenced. So what we did was we first asked them to basically send us as many tissues as possible from each of these 3 individuals. So there's actually kind of 5 individuals kind of split into these three categories. So the M1 and M2 individuals were the high quality fresh frozen, the 09 and 05 were the ones that were a little dead, and the 008 individual was the one that was really, really dead. So first we extracted the M1 and M2 individual, and you can see from here, from the heart and the liver tissues that this DNA is really high quality. You can see the DNA is mostly bigger than 50 kb, streaking up into many, many hundreds of kb. You can see with the second individual, which was kind of dead, but actually preserved in RNA later, you can see that his DNA was actually, still pretty high quality, but it was a little smaller. You can see that it streaks from about 50 kb up to a few hundred kb, but doesn't have all that streaking up in the top end. And you can see more streaking at the lower end here. And then you can see the last individual, the 008 individual, the heart tissue is here, the liver tissue is here. You can see that DNA is very degraded, right? You can see most of the DNA is less than 50 kb here and here. And then you can see there's a lot of pileup at the bottom of the gel from degraded DNA. So basically, we picked the three best samples, which are kind of highlighted with these pink squares. The heart sample from the fresh frozen, the kidney from the kind of dead individual, and the heart sample from the very dead individual. And these were sequenced on PacBio CLR. So this was before kind of HiFi kind of took off. And what we see is that the DNA quality or the sequencing data kind of performance correlates very strongly and directly with the sample quality that was going in. So the heart sample from the fresh frozen individual had a very nice kind of subread and read length N50, about 35 kb. The kind of dead sample, the kidney sample, had a shorter subread N50, but still decent subread N50 of 24 kb. And the really dead individual had a very short subread N50 of 5 kb. So it's quite low, but it's still usable. So basically here you can see 2 things. both, in order to get nice quality sequencing results, the extractions have to be good, but also the tissue going into the extractions has to be well. So it has to be well preserved, well taken care of. And there's also a big tissue type difference too. So, if you have a sample that's a little old, slightly less good, those different tissues are going to degrade at different rates. What we see is that liver tissue degrades very, very quickly. So that really needs to be really quite fresh in order to generate good sequencing results. But we often see that other tissues, such as heart and kidney, can be a little more tolerant of some slight degradation. Recently, a few months later, about 6 months later, that group came back to again, and they wanted to sequence a fourth full sample. This again was a high quality sample. Fresh frozen tissue is available. So again, we asked them for 4 different tissue types. We had liver, spleen, skeletal muscle, and kidney. You can see from the gel and from the pulse field data on the right here that all of these generated quite nice extraction results, good purities, good yields, good DNA size. So in the end, they actually chose to sequence the kidney sample. This time that got sequenced on PacBio Hi-Fi. So in here, they're reading the same molecule, over and over to build high consensus accuracy. They also at the same time resequenced the whole heart sample from before. And what you can see here is that basically all of these samples work very nicely. They all had nice median read quality, QV scores in about 30, generated about 20 to 30 gigs of Hi-Fi CCS data. And basically this all worked quite nicely. So here, we've kind of seen how basically, different input tissue qualities and different treatment methods, whether it's fresh frozen, different tissue types, whether it's degraded, trap caught, RNA later preserved, how these can basically be dealt with to kind of improve the sequencing results, or to basically try to get the best that you can do with what conditions you have. So for the second half of the talk, I'm going to switch gears a little bit and go from these samples, which kind of vary from good to slightly compromised, to what you can do when you have a really high quality sample. So another project that we've been working along on is kind of ultra long sequencing on Oxford Nanopore. So this is where we're trying to generate reads that are hundreds of kb to megabase in length. And the pioneer really kind of in this, one of the pioneers in this area was Josh Quick from Nick Lohman's lab at University of Birmingham. And he basically came up with this protocol that's based on Oxford Nanopore's rapid sequencing kit. So this kit uses transposase to generate the libraries for sequencing on Nanopore. And he modified that protocol by basically greatly increasing the amount of DNA going in, and then reducing the amount of transposase that's used to basically minimize the number of cuts per DNA molecule. So ideally, you get one cut per molecule, and that enables you to kind of preserve your DNA length and then go on to the sequencer. So this method works well. It was what was used to generate a lot of the Nanopore sequencing data in Karen's T2T work, but it has a few challenges. So the first challenge is that the throughput is generally fairly low. So when you run it on MinION, you generally get that should be 1 gigabase, not a megabase. So you generally get about a gigabase per flow cell, and that's a challenge, because if you want to do larger genomes and don't want to get sufficient coverage, you have to run a lot of flow cells. So basically, when we started this project about 1 or 2 years ago, we wanted to see what we could do to try to improve that throughput and those read lengths. So basically, we took a soup to nuts approach and changed everything from the second the sample goes in to the library prep to the sequencing. So we redid the extraction, optimized library prep, and then also took a closer look at the sequencing. The first kind of result of that was we basically, the high molecular weight DNA that you're working with here is generally going in at a very high DNA concentration. It's about a microgram per microliter. You can imagine when the DNA is that big and that concentrated, it's very, very viscous. And that makes it very difficult to handle. It makes the reactions challenging. So we basically took our extractions and we modified them to be able to generate DNA that was less viscous. And then we also modified the library prep too to kind of deal with this challenge. The third challenge with this protocol is that when you take libraries generated with this original method and run them on Prometheon, you generally don't see a bump up in throughput that you expect. Also, when you use the nuclease flush strategy, which is basically when you try to load multiple libraries onto the same flow cell to increase the overall throughput, you don't see that the throughput increases as that's expected either. So to address this, we also made some library prep changes to make both of these things work better. So on the extraction side, this is kind of just a very high-level picture of how our extractions work. They're very simple. There's basically a lysis step. You bind to the magnetic disk, you wash, and then you elute. So basically what we did was we modified our extraction chemistries and the protocols themselves a little bit to be able to reduce the viscosity of that megabase-sized DNA without affecting the size. Here we have some Biorad gels. This is basically the only way that you can use to look at DNA this large. So basically what we want to see is that DNA has large amounts of megabase-sized DNA, and that as much of it as possible is greater than, say, 500 kb. If you use standard high molecular weight DNA, which is kind of less than, let's say, 300 kb, that's really too small to generate ideal results on this protocol. So basically, we have pulse field gels from a couple cell lines and from E. coli and blood. And what you can see is we see a lot of smearing from 50 kb up to base range. But most importantly, we see this compression zone banding that generally tells you that there's a lot of megabase DNA in these samples. So the second thing that I did, or that we did in collaboration with Oxford Nanopore, was we took a close look at the library prep. So the first thing that we did was we went in with a much more dilute solution of DNA. So now you can see that the concentration of DNA in the library prep is generally about 100 nanograms per microliter instead of 1 microgram per microliter. Second, we also tweaked the reaction chemistries during the library prep itself to kind of increase the efficiencies that everything was happening at. And third, we introduced, and most importantly, kind of a nanobind reaction cleanup that enables all of this to work efficiently together. So much like you have Ampere in your standard ligation preps, we're now performing a cleanup at the very end using Nanobind. But the Nanobind process is actually much more gentle than an Ampere process. So we're able to do purifications without shearing that ultra long DNA. The final change that we did was we're actually building a single 3x library that we're loading kind of 3 times onto the same flow cell. And then in between those libraries, we're doing nuclease flush to wash out the old library. So the net effect of all these changes is that we've been able to really increase the pore occupancy. With the original ultra long protocol, the pore occupancies were often quite low. You might see pore occupancies of a few percent, which means that most of the pores aren't actually sequencing. Now we're routinely able to see pore occupancies that are greater than 75%. Second, we've been able to get this protocol to scale efficiently on Promethean. So now if you take the same library and run it on MinION versus Promethean, you'll generally see a 3 to 4-fold higher throughput on Promethean compared to MinION. And finally, we've been able to kind of effectively incorporate this nuclease flush strategy. So now with 3 libraries, we're able to get about 2 to 3x increase in throughput compared to just loading a single library. When you add all this together, basically the net effect is now we're able to get 10 to 100 times higher throughputs than you could with the previous protocol. So on this page, we have a couple examples of the throughputs that we typically see on MinION versus Promethean. On MinION, we're usually seeing somewhere in the 10 to 20 gigabase range. 20, that's 10 to 20 gigabase is pretty typical. 20 to 22, 24 is about the max that we've seen. On Promethean, we're typically seeing in the 40 to 60 gigabase range. About 60 gigabase is the upper end of that. Here you can see the throughput that's generated as a function of time for each of those libraries. And you can see that we generate about a third of the total throughput per load. And here we have a couple of read length distributions that came from basically we generated one large library, sequenced that same library on MinION versus Promethean. And you can see that basically these 2 libraries look almost identical. So you're able to run the same library, the same workflow, and then just decide at the very end whether you want that to run on MinION versus Promethean, and you'll get the same results, except on Promethean, you'll get about 3 to 4 times more data. It's important to note that compared to kind of previous ultra long protocols where we optimize for super high N50, what we found here is that in these protocols, what we're optimizing is actually the absolute amount of data in this blue versus pink bucket. So basically, we want to optimize for the absolute number of reads that are greater than 100 and 200 kb. And we found that the best way to get that is actually to target a slightly lower N50 where we can keep the pores fully occupied. So now you can see that on Prometheon in this run where we generated 60 gigabase total, over 20 gigabase of that was in reads greater than 100 KB, and over 8 gigabase of that was in reads greater than 200 KB. So one of the first places we've applied this protocol was we collaborated with UC Santa Cruz and Oxford Nanopore to apply this to the Human Pan-Genome program. We worked with UC Santa Cruz to develop a nanopore pipeline such that they could apply this to the first 30 genomes in their pilot set. That pipeline basically consisted of 3 Prometheon flow cells with a 3x library per flow cell. And here you can see the results from the first 18 genomes of that. All 30 have been completed, but this is just the first 18 genomes. So you can see that on average, they got about 30x total coverage from those flow cells, 9x total coverage and reads greater than 100 KB, and about 3x coverage and reads greater than 200 KB. So these sequencing results were actually generated about 3 months ago, but you can see that in those last 3 months, compared to the previous data set that I showed you, that we can now generate this data in almost one flow cell. So this protocol is definitely rapidly improving. And that's really what this next slide is about. So I think we're still quite far away from reaching kind of maximum performance limitation of this protocol. So the longest read generated to date has been a 4 megabase read that was generated internally by Oxford Nanopore. Earlier this year, and in the beginning of the year, we generated a 2.4 megabase read that broke Matt Luce's record of 2.2 megabase. And then a few months later, we generated a 2.9 megabase read. And then 1 week after that, Nanopore beat our record and generated this 4 megabase read. So we have no doubt that once we release this protocol kind of into the wild and customers are able to kind of apply this, that we're going to see some monstrous reads and new records generated quite often. So as I was putting these slides together, ONT actually generated more data that actually kind of outdated just that previous data set that we generated just a month ago. So the last data they generated almost 80 gigabases of total data at 128 KB and 50. You see that from this run, almost 50 gigs was in reads greater than 100 KB, and almost 22 gigs was in reads greater than 200 KB. So this is really some pretty monstrous ultra long data. And then just yesterday, they told us that they'd actually generated an even longer run. So they'd broken what they call the double century mark and generated a run that had 100 plus gigs of data at 100 plus KB and 50. So as you can see, we're getting to the point that we can generate ultra long genomes from a single flow cell, basically of a human sized genome. So the last part would be basically, to run this new protocol, you're gonna need a few kits and components from us and from ONT. You'll need the Nanobind Big DNA kit, one version of that to extract this megabase DNA. There'll be an ultra long library prep kit that's going to be released shortly to do those reaction purifications, and there'll be a new sequencing kit from ONT to be able to prep those libraries. At the same time, we'll be releasing protocols for cells, bacteria, and blood, and also for tissue samples. There also will be new sequencing scripts released to reduce and eliminate the read splitting that can occur. And we'll also be releasing data sets of ultra long data that you guys can download and play with to see what this ultra long data looks like. And all of that should be coming 12/1 at the ONT Nanopore community meeting. So we'd like to kind of thank all our collaborators. For the Nanopore work, we work closely with Oxford and with UC Santa Cruz. For the PacBio work, we worked closely with Polly Campbell and Brian Cougar, and all of that sequencing was done at University of Maryland with Luke Talon. A lot of this work was funded by the very awesome SBIR program at NIH. They funded a lot of our research and development, and we're eternally grateful to them. Thanks.
All right, thank you, Kelvin. So now we'll open up to some questions here. We had a couple of questions about different kits that Circulomics might be developing for viruses and potentially metagenomics. What's the status on those?
Yeah, so we're kind of in the early phases of developing a metagenomic kit. You know, I guess with that kit, one of the challenges is always bias and representation. You know, how to maintain a uniform representation across things that are tough to lyse versus easy to lyse, and then balancing that was getting nice high quality DNA out of it. So we're kind of in the early phases of working on that. Hopefully, we'll have something available for testing in 2021, but we don't have a hard date identified for that yet.
Okay, and then so the next one is with the latest ultra long data that you showed. Could you clarify? Did that use the ultra high molecular weight extractions and the UL library prep together?
Yes, yeah. So that requires both of those things. So we actually have new ultra high molecular weight DNA extraction protocols that we'll be releasing at the same time as the library prep protocol. So you'll need to do the Nanobind ultra high extractions at the beginning to get the very large DNA, and then you'll need to purify it during the library prep with the second library prep kit.
Okay. So let's see, we have some time for some few more questions here. So what is the Nanobind purification efficiency?
I guess that question is probably asking for the recovery efficiency. I'm not sure if they're asking for that in the library prep or in the extractions themselves.
Yeah, maybe you could address both.
Yeah, so in the extractions themselves, we think they're generally quite high. So we think generally that should be greater than 80%. So we generally should be getting back most of the DNA that's in there. For the library prep kits, it should be quite high. It should be quite high too. When you're running the ultra long library prep kits, once that's eventually released, it's actually a little bit difficult to quantify accurately that DNA because you'll find that it is quite heterogeneous because it's so big. But the recovery efficiency should be fairly high. We generally think that they're greater than 80%.
Okay. And then we have a question here. What is the volume required to fully submerge the Nanobind discs?
Yeah, so the volume that's needed to submerge the Nanobind discs it varies a bit depending on what size of disc is used and the kind of shape of the tube that it's in. So that's kind of why we offer discs of all different sizes. So for the standard kind of 5mm disc, you generally need about 50 to 100 microliters if it's in a standard 1.5 mil microcentrifuge tube. If you flip it over in the cap, it can be less. When we're doing very small volume extractions, oftentimes we use a small 1 or 2 millimeter disc, and for that you only need a 5 or 10 microliter volume. So it can be kind of scaled based on the size of the disc, and that's one thing that we play around with for these different sample types and protocols.
All right, and then we have a question here about what correlation do you see between the size distribution and the loading amount?
Between the size distribution and the loading amount. So the loading amount should generally not affect the size distribution. That would mainly affect the amount of throughput that you get, but the amount of DNA going into the prep is quite important. So for these preps, we're generally going in with we're generally going with about 50 micrograms of total DNA going in the front end. If you put in less DNA, what happens is that those reads will start to get shorter, but your throughputs will actually start to go up. So we've gone down to, let's say 30 micrograms. It works fairly well with the 3x library. If you're willing to go down to a 1x library, that could again be decreased. So that's one thing that we'll be actively looking at with kind of version 2 of the protocol, would be kind of decreasing the input requirements for this protocol.
Okay, I think in order to stay on time, I think that's where we'll wrap things up. Thanks again, Kelvin. That was a great talk.